body fluid and circulation. Simple organisms like sponges and polypterats circulate water from their surroundings through their entire body cavities to facilitate the cells to exchange of these substances. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also helps in the transport of certain substances. Blood. Blood is a special connective tissue consisting of a fluid matrix, plasma, and formed elements. Plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. Fibrinogen. Fibrinogen, globulins and albumins are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanisms of the body. Albumins help in osmotic balance. Plasma also contains small amounts of minerals like calcium plus, C plus plus, Mg plus plus, SU3 plus, minus, Cl minus, etc. Glucose, amino acids, lipids, etc. are also present in plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factors is called serum. Formed elements, erythrocytes, leukocytes, and platelets are collectively called formed elements and they constitute nearly 45% of the blood. Erythrocytes, RBC, are the most abundant of all these cells in blood. A healthy adult man has on average 5 millions to 5.5 millions of RBCs per cube of blood. RBCs are formed in the red bone marrow in the adults. RBCs are devoid of nucleus in most of the mammals and biconcave in cells. They have a red-colored iron-containing complex protein called hemoglobin. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, graveyard of RBCs. Leukocytes, WBCs, is they are colorless due to the lack of hemoglobin. WBCs are nucleated and relatively larger in number, which averages 6,000 to 8,000 per cube of blood. Leukocytes are generally short-lived. Two main categories of WBCs are granulocytes and agranulocytes. Granulocytes are also divided into three types: neutrophils, eosinophils, basophils. And agranulocytes are also divided in two types: lymphocytes, monocytes. Neutrophils are the most abundant cells, 60 to 65% of the total WBCs. Basophils are the least, 0.5% to 1%. Neutrophils and monocytes, 6 to 8%, are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Eosinophil, 2 to 3%, resist infections and are also associated with allergic reactions. Lymphocytes, 20 to 25%, are of two major types, B and C. Forms both B and T lymphocytes are responsible for immune responses of the body. Platelets, thrombocytes are cell fragments produced from megakaryocytes, special cells in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per cube. Platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorder, which will lead to excessive loss of blood from the body. Blood grouping. ABO grouping is based on the pressure or absence of two surfaces, antigens, chemicals that can induce immune responses on the RBCs, namely A and B. Blood grouping, antigen, antibodies in plasma, donors group. A group antigen on RBCs A, antibodies in plasma anti-B. Donors group A and O. B blood group, antigens on RBCs B, antibodies in plasma anti-A. Donors groups B and O. And blood group AB, antigens on RBCs A and B, antibodies in plasma nil. Donors group A, B, and O. O blood group, antigens on RBCs nil, antibodies in plasma anti-A and anti-B. Donors group only O. Universal donors O. Universal recipients AB. Rh grouping. Another antigen, the Rh antigen, similar to one present in rhesus monkeys. Hence, Rh is also observed on the surface of RBCs of majority, nearly 80% of humans. An average positive is exposed to Rh positive blood will form specific antibodies against the Rh antigens. Erythroblastosis. Erythroblastosis fertility. A special case of Rh incompatibility has been observed between the Rh blood of a pregnant mother with Rh positive Rh negative blood of a pregnant mother with the Rh positive blood of the fetus. Rh antigens of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to small amounts of the Rh positive blood from the fetus. In such cases, the mother starts repairing antibodies Rh antigen in her blood. In cases of her subsequent pregnancies, the Rh antibodies from the mother Rh negative can leak into the blood of the fetus Rh positive and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice of the baby. This can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. Coagulation of blood. A dark reddish brown scum formed at the site of a cut or an injury over a period of time. It is a clot or coagulation formed mainly of a network of threads called fibrins, in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the convergence of inactive fibrinogens in the plasma by the enzymes thrombin. Thrombins in turn are formed from another inactive substance present in the plasma called prothrombin. An enzyme complex thrombokinase is required for the autoreaction. Cascade process. This complex is formed by a series of linked enzyme reactions. Cascade process involving a number of factors present in the plasma in an inactive state. Calcium ions play a very important role in clotting. Lymph tissue fluid. As the blood passes through the capillaries in tissue, some water along with many small water-soluble substances move out into the spaces between the cells of tissues, leaving the larger proteins and most of the formed elements in the blood vessels. This fluid released out is called the interstitial fluid or tissue fluid lymph. An elaborate network of vessels called the lymphatic system collects the fluid and drains it back to the major veins. The fluid present in the lymphatic system is called the lymph. Lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the reticules present in the interstitial villi. Circulatory pathway. The circulatory patterns are of two types, open and closed. Open circulatory system is present in arthropods and mollusks in which blood pumped by the heart passes through large vessels into open spaces or body cavities called sinuses. Annelids and chordates have closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of blood can be more precisely regulated. All vertebrate possess a muscular chamber heart. Fishes have the two-chambered heart with an atrium and a ventricle. Amphibians and the reptiles, except crocodiles, have a three-chambered heart with two atria and a single ventricle. Crocodiles, birds, and mammals possess a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood, which is oxygenated by the gills and supplied to the body parts, from where deoxygenated blood is returned to the heart single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills, lungs, skin, and the right atrium gets the deoxygenated blood from other body parts. However, they get mixed up in the single ventricle, which pump out mixed blood in complete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria relatively passes on to the ventricle of the same size. The ventricles pump it out without any mixing of its two separate circulatory systems. Separate pathways are present in the organism's double circulatory system. Human circulatory system. Heart, the mesodermally derived organ, is situated in the thoracic cavity in between two lungs, slightly lateral to the left. Heart has the size of a clenched fist. Each heart is protected by a double wall membranous bag, pericardium, enclosing the pericardial fluid. Heart has four chambers, two relatively small upper chamber and colectia, and two large lower chamber called ventricles. A thin muscular wall called the inner atrial septum separates the right and the left atria, whereas a thick wall atrial septum separates the right and the left ventricles. The atrium and the ventricle of the same side are also separated by a thicker fibrous tissue called the atrioventricular septum. However, each of these septa are provides with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a wall formed of three muscular flaps of the tricuspid valve, whereas the bicuspid or mitral valve guards the opening between the left atrium and the left ventricle. The opening of the right and the left ventricles into the pulmonary artery and the aorta respectively are provided with the semilunar valves. These valves in the heart allows the flow of blood only in one direction, from the atria to the ventricles and from the ventricles to the pulmonary artery or aorta. This valve prevents any backward flow. Heart. The entire heart is made up of cardiac muscle. The walls of ventricles are much thicker than that of atria. A specialized cardiac musculature called the nodal tissue is also distributed in the heart. A patch of nodal tissue is presented in the right upper corner of the right atrium called the sinoatrial node (SAN). Another mass of nodal tissue is seen in the lower left corner of the right atrium close to the anteroventricular septum called the anteroventricular node (AVN). A bundle of nodal fibers, atria ventricular bundle (AV bundle), continues from the AVN, which passes through the anteroventricular septa to emerge on the top of the interventricular septum and immediately divides into right and left bundle. These branches give rise to thinner fibers throughout the ventricular musculature of the respective sides and are called Purkinje fibers.